Right now, inside your body, there are microscopic clusters of cancer cells. They have been forming and dissolving your entire life, completely without your knowledge. And the reason you are sitting here, alive and breathing, is because your body has been winning that silent war over and over again. Most people do not know that. Most doctors do not even talk about it this way. But it is one of the most profound and empowering truths in all of modern medicine. Here is what I mean. Researchers have done autopsies on people who died from completely unrelated causes, car accidents, heart attacks, things that had nothing to do with cancer, and what they found in the tissue samples was staggering. Virtually every single person over the age of 40 had microscopic cancers hidden in their body, tiny clusters of abnormal cells sitting silently in the thyroid, in the breast, in the prostate. They were there, but they never grew. They never became a problem. Why? Because of one biological process, one mechanism that most people have never heard of. And once you understand it, food will never look the same to you again. That process is called angiogenesis. Angiogenesis. It sounds complicated, but the concept is so simple and so elegant that once I explain it, you will wonder why every person on this planet is not talking about it every single day. Angiogenesis means the growth of new blood vessels, the body's ability to sprout brand new blood vessels from existing ones. It happens all the time, and it is essential to life itself. When you get a cut on your finger, angiogenesis helps heal it. When a pregnant woman grows a placenta, angiogenesis builds the network that feeds the baby. It is a normal, healthy, miraculous process. But here's the thing, cancer figured out how to hijack it. Now think about that for a second. A tiny cluster of cancer cells, something smaller than the tip of a ballpoint pen, that cluster desperately needs blood. It needs oxygen. It needs nutrients. Without its own blood supply, it literally cannot grow beyond a certain size. It just sits there, dormant, harmless. Scientists call these dormant clusters micrometastases, and in their tiny prevascular state, they cannot kill you. They can coexist inside your body for decades. Your immune system keeps them quiet. Your body keeps the balance. But if those cancer cells successfully recruit blood vessels, everything changes. The signal they use has a name, VEGF, vascular endothelial growth factor. Think of it as a distress flare sent up by a cancer cell into the surrounding tissue. It is saying, over here, send blood, send oxygen. We are ready to grow. And if the body responds to that flare, you get what scientists call the angiogenic switch. The moment that switch is flipped, the cancer goes from harmless bystander to aggressive invader. Within days, not weeks, not months, days, of gaining its first blood vessel, a tumor can double and double again. The supply line is open, the highway is built, and now the cancer has everything it needs to invade neighboring tissue and send rogue cells streaming through your bloodstream. This is why angiogenesis is not just a scientific curiosity. This is the battlefield. This is where the war is actually fought. Now here is the part that most people miss completely. Your body is not passive in this battle, not at all. Your body has a natural built-in system of anti-angiogenic defenses, molecules and proteins that patrol your tissues, constantly looking for runaway blood vessel growth and constantly suppressing it. When your anti-angiogenic system is robust and working properly, those tiny cancer clusters stay tiny. The signal is sent, but no one answers. The flare goes up, but the blood vessels do not come. The cancer starves before it ever has a chance to breathe, but here is the fascinating part. The food you eat every single day can either strengthen or weaken that anti-angiogenic defense system. Every meal is a conversation with your biology. Every bite is either sending your defenses to the front lines or pulling them back. And the science on this is not speculative. It is published in journals like Nature, Cell, and The Lancet. Let me take you inside the science, because this is where it gets genuinely extraordinary. Imagine your anti-angiogenic defense system as a network of gates on a highway. A healthy body keeps those gates closed. It controls when and where blood vessels are allowed to grow. Cancer tries to pick the locks, and certain molecules in food act like reinforcements for those gates. They make the locks unpickable. The first compound I want to discuss is one that billions of people consume every single day, often without realizing the profound biological effect it is having. It is EGCG, Epigallocatechin 3 gelat, found in green tea. When you drink a cup of properly steeped green tea, the EGCG molecules are absorbed from your gut into your bloodstream within 30 to 60 minutes. 30 to 60 minutes. This is not slow. This is not gradual. This is rapid biological action. Those EGCG molecules travel through your circulation and begin interacting directly with VEGF receptors on the surface of endothelial cells, the cells that line your blood vessels. They block those receptors from receiving the cancer cell's distress signal. They jam the frequency. Think of it this way. The cancer cell is trying to call in reinforcements using a specific radio frequency. EGCG comes in and jams that channel. The reinforcements, the blood vessels, never receive the call. They never arrive. The cancer stays hungry. This has been demonstrated in laboratory studies, in animal models, and increasingly in human clinical data. At the University of Wisconsin, researchers found that EGCG inhibited the formation of new blood vessels in tumor models with a potency that rivaled some pharmaceutical anti-angiogenic agents. A cup of tea, a natural molecule, performing comparably, in specific contexts, to a drug. The science on this is real, but EGCG does not just jam the signal, it goes a step further. It actively promotes the production of a protein called thrombospondin 1, which is one of your body's own natural anti-angiogenic molecules. So EGCG is simultaneously blocking cancer's communication line and amplifying your body's own defensive response. It is working on two fronts at the same time. The dose matters here. Studies suggest 2 to 4 cups of green tea per day, providing roughly 200 to 400 milligrams of EGCG, begins to produce measurable biological effects. Not a pharmaceutical, not a megadose supplement. Tea you can make in your kitchen this afternoon. And here is a detail most people do not know. Adding lemon juice to your green tea, because of its vitamin C content, significantly enhances EGCG absorption by protecting it from oxidation in the gut. Lemon in your green tea is not just a flavor preference, it is a strategy. Now let me tell you what EGCG is doing at an even deeper molecular level, because I want you to appreciate just how sophisticated this natural compound truly is. EGCG also inhibits matrix metalloproteinases, enzymes that cancer cells use to literally chew through the extracellular matrix surrounding them, creating space for new blood vessels to invade. Think of the extracellular matrix as the scaffolding of your tissues. Cancer uses these MMP enzymes like a chainsaw to tear through that scaffolding. EGCG blunts those chainsaws. It inhibits MMP2 and MMP9, two of the most critical enzymes in tumor invasion. Without those enzymes working at full capacity, the cancer cannot clear a path for blood vessel growth. And simultaneously, EGCG activates pathways inside endothelial cells that promote a form of programmed cell death in cells that have detached and are migrating toward a tumor. The supply line builders are made to turn against themselves, right in the middle of their journey. This happens within 2 to 3 hours of consumption. 2 to 3 hours. Think about a morning routine that includes green tea with lemon. The anti-angiogenic reinforcements are being deployed before you even sit down at your desk. Now let's move to the second compound, and for this one, the way you prepare this food actually determines its potency. I am talking about lycopene, found in tomatoes. Here is a fact that genuinely surprises people. Raw tomatoes contain lycopene, yes, but when you cook a tomato with a small amount of healthy fat, olive oil for instance, the heat breaks down the cell walls, and the bioavailability of the lycopene increases by as much as 2 to 300%. Cooking the tomato makes it dramatically more powerful. This is the opposite of what most people assume. Most people think raw is always better, but with lycopene, heat is your friend. A slow-cooked tomato sauce, a thick marinara, a roasted tomato with olive oil. These are not just delicious. They are biological upgrades. So what does lycopene actually do once inside you? It is fat-soluble, which is why the olive oil matters. It helps with absorption. Once absorbed, lycopene accumulates in specific tissues, notably the prostate in men, breast tissue, and the liver. It builds up with regular consumption. And in those tissues, lycopene interferes with a critical step in angiogenesis, the step where endothelial cells actually migrate and multiply toward the tumor. For a blood vessel to grow toward a cancer cell, the cells that make up the vessel wall have to move. They have to proliferate. They have to build the road, brick by brick. Lycopene specifically inhibits the proliferation and migration of those endothelial cells. It freezes the road-building crew in place. The cancer sends the distress signal, the crew shows up at the work site, and then they just stop. They cannot lay the track. The highway to the tumor never gets built. Harvard researchers published extraordinary data from the Health Professionals Follow-Up Study, a massive, long-running investigation involving tens of thousands of men, showing that those who consumed the highest amounts of cooked tomato products had a significantly reduced risk of prostate cancer. Not a small reduction, a statistically robust, clinically meaningful reduction, and the association was strongest with cooked tomatoes, not raw ones, which perfectly aligns with the bioavailability science. Cooking tomatoes with olive oil is not just a Mediterranean tradition. It is a biological strategy that has been quietly protecting people for centuries without them knowing why. Now let me tell you about the third compound, and this one is extraordinary in its elegance. I'm talking about ellagic acid and its gut-derived metabolites called urolithins, found in pomegranates, strawberries, raspberries, and walnuts. Here is where it gets really interesting. Ellagic acid in these foods is itself anti-angiogenic. It directly inhibits VEGF-induced blood vessel formation. But ellagic acid is not the end of the story. It is a precursor. When you eat ellagic acid-rich foods, the bacteria in your gut, your microbiome, begin metabolizing that ellagic acid and transforming it into compounds called urolithins, specifically urolithin A and urolithin B, and these metabolites are even more potent anti-angiogenic agents than the original compound. Your gut microbiome is acting as a biochemical refinery. It takes raw material from your food and converts it into something more powerful. Your body is upgrading the weapon. Now, listen closely to this. The effectiveness of ellagic acid-rich foods in any individual depends partly on the composition of their gut bacteria. Some people produce urolithins efficiently, others do not, at least not as readily. But the foods that promote a healthy microbiome, fermented foods, fiber-rich vegetables, diverse plant eating, also create the conditions in your gut that favor urolithin production. So it is another example of synergy, your dietary patterns working together across multiple biological systems toward the same anti-cancer goal. In laboratory studies at the University of California, researchers demonstrated that urolithin A suppressed the growth of new blood vessels in tumor tissue with measurable potency. And pomegranate juice, whole, unsweetened pomegranate juice, showed anti-angiogenic activity in cell studies at concentrations achievable through normal dietary consumption. These are not theoretical doses. These are amounts you can actually drink. I want to pause here for a moment, because something important needs to be said clearly. I am not telling you that eating berries cures cancer. I am not telling you to stop your cancer treatments or ignore your oncologist. Please hear me on this. What I am saying is that the foods you eat create a biochemical environment inside your body, and that environment either supports your body's own cancer defense systems or undermines them. And the science on this is real, peer-reviewed, and increasingly being integrated into clinical oncology at places like Memorial Sloan Kettering, MD Anderson, and Dana-Farber. Because they understand what I am describing. Angiogenesis is the mechanism. Food-derived bioactives are the natural modulators, and your body is the battlefield where this is being won or lost, one meal at a time. Now I want to bring in one more compound, because it would be a significant omission to leave it out. I am talking about resveratrol, found in red grapes, red wine, peanuts, and certain berries. Plants make resveratrol when they are under stress, when a vine is attacked by a fungus or exposed to UV radiation. It is a biological alarm response encoded in plant chemistry. And when we consume that plant, we absorb that molecular defense signal and it activates our own defense pathways. This concept has a name in science, xenohormesis, the idea that stress response chemicals made by plants can activate stress resilience pathways in the animals that eat them. The plant's struggle becomes our strength. Resveratrol's anti-angiogenic mechanism targets a transcription factor called HIF-1 alpha, hypoxia-inducible factor 1 alpha. This is, in a sense, the master switch of angiogenesis. When a tumor outgrows its blood supply and becomes low in oxygen, HIF-1 alpha is activated, and it cranks up the production of VEGF and dozens of other pro-angiogenic factors all at once. HIF-1 alpha is the cancer's emergency broadcast system. Resveratrol turns down the volume on that broadcast. By suppressing HIF-1 alpha activity, resveratrol reduces the overall angiogenic output of a hypoxic tumor. It does not just block one signal, it suppresses the signal-generating machinery itself. It goes upstream and attacks the problem at its source. One glass of red wine with dinner, a small serving of red grapes, a handful of peanuts. These are not exotic interventions. They are accessible, pleasurable, and biologically potent. And let me now bring in one final compound that most conversations about cancer-fighting foods completely overlook: sulforaphane, found in broccoli, and especially in broccoli sprouts, which contain 50 to 100 times the concentration found in mature broccoli. Sulforaphane activates a master regulatory protein called NRF2, which controls the expression of hundreds of genes involved in antioxidant defense, detoxification, and notably anti-angiogenic signaling. When sulforaphane enters your cells and activates NRF2, it is like turning on a control panel that regulates hundreds of protective genes simultaneously. It is upstream regulation of your entire defensive infrastructure. Researchers at Johns Hopkins found that broccoli sprouts specifically inhibited tumor angiogenesis in animal models and showed biological effects in human tissue within hours of consumption. Hours. The response is not slow, it is rapid and measurable. And here is the beautiful irony. Broccoli sprouts are cheap. You can grow them at home in a jar on your kitchen counter in 3 to 5 days. You can produce one of the most potent anti-angiogenic food sources known to science on your windowsill for almost nothing. The democratizing power of that is something I find genuinely moving. You do not need a prescription. You do not need a specialist. You need a jar, some water, and broccoli seeds. Now, I said earlier that most discussions about cancer-fighting foods overlook one critical element, and that element is timing. Everyone talks about what to eat. Almost nobody talks about when to eat. And the biological timing turns out to be deeply relevant to anti-angiogenic defense. There is growing research around time-restricted eating and its effects on tumor angiogenesis. When you compress your eating into a window of 8 to 10 hours and give your body a sustained overnight fast of 14 to 16 hours, something remarkable happens. During those fasting hours, insulin levels drop. And this matters enormously because insulin is a potent pro-angiogenic signal. High insulin levels promote VEGF expression. They create a biological climate that is friendly to blood vessel growth. A sustained fast, by bringing insulin down, creates a climate that is hostile to angiogenesis. Simultaneously, fasting activates autophagy, your cells consuming and recycling their own damaged components. Autophagy is part of your body's quality control system, and it plays a significant role in identifying and eliminating precancerous cells before they ever reach the angiogenic switch. So the overnight fast is not empty time. It is active, defensive, biological housekeeping, and simply shifting dinner earlier and breaking fast a little later can meaningfully extend that protective window. Let me now pull everything together, because I want this to crystallize clearly in your mind. Your body has cancer in it right now, almost certainly. Tiny, microscopic clusters of abnormal cells kept dormant and starved by your anti-angiogenic defense system. Your single most important job biologically is to keep those defenses strong. EGCG from green tea jams the cancer's communication signal and blunts the enzymes it needs to clear a path for blood vessel invasion. Lycopene from cooked tomatoes freezes the endothelial cells that would otherwise build the highway toward a tumor. Ellagic acid and its gut-derived urolithins attack multiple overlapping communication channels in the angiogenic cascade. Resveratrol goes upstream and turns down the master alarm system the cancer uses to call for help, and sulforaphane activates a control panel of hundreds of protective genes simultaneously. These are not isolated effects working independently. They are interlocking pieces of the same defensive architecture, and together they create a biological environment inside your body that is profoundly hostile to the angiogenic process that cancer depends on. This is what researchers call multi-targeting. Pharmaceutical drugs typically hit one molecular target. A diverse diet, rich in bioactives, simultaneously hits dozens of overlapping targets in the same pathway. And cancer, which is notoriously good at mutating around single-agent therapies, finds it extraordinarily difficult to escape a multi-flanked siege. You are not giving cancer one lock to pick. you are giving it 20. Now, I want to be honest about the limits of what we know. Most of the human data on dietary anti-angiogenesis is epidemiological. Population studies tracking what people eat and what cancers they develop. This type of research shows powerful associations, but associations are not the same as proven, isolated causation. Randomized controlled trials are harder to conduct with food than with drugs. You cannot give someone a placebo tomato. The effects of diet operate over decades, which makes controlled trials enormously complex. So what we have is a convergence of evidence, epidemiological associations, precise molecular mechanisms, animal model data, and increasingly early clinical trial signals, all pointing in the same direction from independent research groups around the world. That convergence is how science builds confidence, not with one perfect study, but with many imperfect studies that all point the same way, and they point consistently toward the same conclusion. The foods I have described today, green tea, cooked tomatoes, pomegranates, berries, red grapes, broccoli sprouts, are associated with lower cancer incidence in population studies and demonstrate clear anti-angiogenic mechanisms in laboratory research. Eating them is not a guarantee, but not eating them means leaving a significant part of your natural defense system chronically under-resourced. I want to share one number with you because it grounds all of this in reality. The World Health Organization estimates that somewhere between 30 and 50% of all cancer cases globally are preventable through lifestyle changes. 30 to 50%. If you could translate that percentage into actual lives, you would be talking about millions of people every single year. Mothers who do not get a diagnosis at 45, fathers who live to see their grandchildren, young people whose lives are not derailed before they have truly begun. And the number one modifiable risk factor in that preventable percentage, ranked even above smoking in some analyses for certain cancer types, is diet. What we put in our mouths 3 times a day, every single day. Let me say something personal before I close, because science without humanity is just data, and data without meaning is just noise. I have sat with patients who were told there was nothing more that could be done. I have seen the look on a person's face when a doctor uses the word terminal. And what I have found in those conversations is that one of the things people grieve most deeply is the feeling that they had no agency, that it just happened to them, that they were powerless. Part of what drives my research into diet and angiogenesis is a deep conviction that this does not have to be a passive experience, that the daily act of choosing what to eat is, in fact, a profound act of biological agency, that every single day, without a doctor's appointment, without a prescription, without a waiting room, you are participating in your own defense. This does not mean perfection. This does not mean living in fear around every meal. It means awareness. It means understanding that food is not just fuel. Food is information. Food is instruction. Food is, in a very real and literal sense, a conversation with your genome, your microbiome, your immune system, and your anti-angiogenic defense network. The conversation happening right now in trillions of your cells today is setting the conditions for what grows and what starves inside your body over the coming years and decades. And change is accessible. Change is in the grocery store. Change is in how long you steep your tea, whether you cook your tomatoes, whether you choose the pomegranate juice over the soda, whether you end dinner an hour earlier and let your overnight fast do its silent, miraculous work. The greatest opportunity in cancer medicine is not in the treatment of established disease. It is in creating a body that never lets the disease establish itself in the first place. That is the future I believe in, a future where understanding your own biology, truly understanding the supply lines and the security forces and the silent wars being fought inside you right now, becomes part of everyday life, where every cup of tea is understood as a biological act, where cooking tomatoes is understood as a defensive strategy, where a handful of berries is understood as an act of genetic self-care, because you have more power over what grows inside your body than anyone ever told you, and the science is there to prove it.